In this week's video, we're going to be talking about muscle, starting with the molecular characteristics of different contractile filaments within a sarcomere. The sarcomere is the functional unit within the skeletal muscle where myofilaments are arranged. The myofilaments are primarily responsible for muscle contraction, are actin, myosin, and titin. When we talk about thick filaments, these are primarily composed of about 300 myosin type 2 molecules bound together surrounding a titin molecule. Each myosin type 2 protein consists of 6 subunits, 2 heavy chains, and 4 light chains. The heavy chains have a long shaft-like structure connected to a globular myosin head. The shafts or tails wrap around each other to form the core of the thick filament. The globular heads project outward at right angles and are capable of binding to active actin sites. The region connecting the head to the shaft is called the hinge region, which can bend and is involved in the power stroke during contraction. Myosin heads also contain an ATPase enzyme that binds to and hydrolyzes ATP, providing energy for contraction. Myosin heads also each myosin head is associated with 2 light chains, which regulate the head's actions. The arrangement of the myosin heads around the thick filament allows each thick filament to interact with 6 thin filaments. When we talk about a thin filament, each thin filament consists of 2 standard fibrous actin or F-actin along with troponin and tropomyosin molecules. F-actin is formed by 2 strings of globular actin or G-actin wound together in a double helix. Each G-actin monomer has a binding site for ATP used during polymerization and later becomes a binding site for myosin, referred to as an active site. Dimers of the protein tropomyosin extend over the F-actin filament and cover the myosin active sites. Other things about the sarcomere, we have Z-disc proteins that form the Z-line borders between adjacent sarcomeres, nebulin that extends toward the center, the elastic protein titin that is found between nebulin molecules and is thought to play a role in resetting the sarcomere after contraction. During muscle contraction, the myosin heads form cross bridges by linking the thick and thin filaments, causing them to slide over each other. This sliding shortens each sarcomere, the muscle fiber, and the entire muscle. The myosin heads have 3 key properties. They bind to G-actin active sites to form cross bridges, and they are connected by the hinge region, and their ATPase activity breaks down ATP to provide energy. Now we're talking about action potentials leading to calcium release and molecular mechanisms of skeletal muscle specifically. So skeletal muscle contraction requires an electrical event or action potential followed by a mechanical event, which is contraction. The sequence begins when an action potential arrives at the axon terminal of a somatic motor neuron. This stimulates the opening of voltage-gated calcium channels in the axon terminal membrane, allowing calcium to enter from the extracellular space. The increased intracellular calcium levels signal the synaptic vesicles containing acetylcholine to undergo exocytosis, releasing that acetylcholine into the synaptic cleft. As it diffuses across the cleft, it'll bind to acetylcholine receptors, which are ligand-gated ion channels, nicotinic type 1, located on the sarcolemma of the muscle fiber at the motor end plate. Binding of acetylcholine opens these ligand-gated sodium-potassium channels. Due to different driving forces, more sodium diffuses into the cell than potassium diffuses out, causing the sarcolemma to depolarize. This depolarization triggers nearby voltage-gated sodium channels to open, initiating an action potential that spreads across the sarcolemma and down to the T-tubules. T-tubules are evaginations of the sarcolemma filled with extracellular fluid. Located at the junctions of the A and I bands, the T-tubules are flanked by dilated regions of the sarcoplasmic reticulum, or the SR from here on out. Called terminal cisternae, forming structures known as triads. The T-tubule membrane at the triad contains voltage-dependent proteins called dihydropyridine, DHP, from here on out, channels. These channels act as voltage sensors rather than actually allowing calcium passage. They are physically linked to ryanodine receptor channels, or RYR, which are calcium release channels on the SR membrane. When the action potential depolarizes the T-tubule membrane, the DHP channel detects this change and causes the linked RYR channel to open. resulting in the release of stored calcium from the SR into the SR terminal cisternae into the sarcoplasm. This allows enough calcium to follow its gradient at release. The moment molecular mechanisms of muscle contraction will then proceed, calcium ions released in the sarcoplasm bind to troponin C, a component of the thin filament. This binding causes a conformational change in troponin, which moves the tropomyosin molecules away from the active sites on the G-actin molecules. With the active sites exposed, the cocked myosin head, bound to ADP and a phosphoryl group, is in a high energy state, can bind to the active sites, forming cross bridges. Upon binding, the phosphate group is released, initiating a power stroke. During the power stroke, the myosin hinge region bends, pulling the actin filament past the myosin filament towards the center of the sarcomere. The ADP is also released from the myosin head during the power stroke. Since myosin heads on opposite ends of the thick filaments pull in opposite directions, the overall effect is to shorten the sarcomere. For continued shortening, the myosin heads must detach from the actin binding sites and reattach further along the actin filament. Binding of a new ATP molecule to the myosin head causes it to detach from actin. The ATPase then hydrolyzes this ATP into ADP and a phosphoryl group, which recocks the myosin head, we call it a recovery stroke, returning to its high energy state and preparing for the next cycle. This cross-bridge cycling continues as long as sufficient calcium is present in the sarcoplasm and ATP is available. Relaxation occurs when calcium is removed from the sarcoplasm. This is primarily achieved by sarcopumps, or sarcoendoplasmic reticulum calcium ATPase pumps, located on the SR membrane, which actively pump calcium back into the SR against its concentration gradient using ATP. The active pumping by Circa is a major consumer of ATP in contracting muscle. As calcium levels in the sarcoplasm decrease, calcium unbinds from the troponin C, allowing tropomyosin to move back and cover the active sites on actin, preventing myosin from binding and terminating the cross-bridge cycle.
These sources state that there are 4 ways muscles obtain their high.
Since ATP is the thing that we use the most to have our muscles contract, the 4 main sources that we get it from is the first is the cytoplasmic stores, which will last about 1 to 5 seconds. Then we have creatine phosphate, which will last about 10 seconds. And then we have our third one is glycolysis, which will last us about 60 seconds. And then we have aerobic respiration, which happens in the mitochondria, which will be able to last us hours. Types of smooth muscles. So smooth muscle is found lining various hollow organs in the body, including the digestive system, airways, urinary system, and blood vessels. While its contraction is slower than skeletal muscle, it can generate more force per area and shorten significantly more. Smooth muscle is also very fatigue resistant. Smooth muscle is broadly categorized into 2 groups. The first one is single unit smooth muscle. So these muscles are linked together by gap junctions, which allow them to contract as a coordinated single unit. Contractions often spread as waves, such as peristalsis in the digestive system. Single unit smooth muscles can generate their own contractions using pacemaker potentials and can function independently of the nervous system, although their activity can be modified by it. The second type is multi-unit smooth muscle. These muscles are not electrically coupled by gap junctions, therefore stimulation of one muscle cell does not directly lead to the stimulation of others. Multi-unit smooth muscles rely solely on nervous or endocrine input for activation. Examples include the iris of the eye and the erector pili muscles. It is noted that the distinction between single unit and multi-unit smooth muscle represents 2 ends of a spectrum, and most smooth muscles are comprised of combinations of the 2. When we talk about mechanisms of smooth muscle contraction, it's initiated by the phosphorylation of the myosin light chain. So unlike skeletal muscle, smooth muscle lacks troponin. Contraction still requires a rise in intracellular calcium, but the mechanisms for increasing intracellular calcium and initiating contraction are different from skeletal muscle. So the first step is to increase intracellular calcium. The smooth muscle cell experiences an increase in intracellular calcium. This calcium can come from the extracellular stores through voltage-gated calcium channels opened by an action potential or through ligand-gated calcium channels opened by a chemical signal. Alternatively, it can come from intracellular stores in the sarcoplasmic reticulum, released via an IP3 second messenger system triggered by a chemical signal. Calcium-induced calcium release through the ryanodine receptor on the SR can also contribute. This ability to use both intracellular and extracellular calcium stores is a key difference from skeletal muscle. The second thing we need to know is that calcium binds to calmodulin. The increase in intracellular calcium ions bind to the intracellular protein called calmodulin, which has 4 calcium binding sites. We also activate something called your MLCK. So the newly formed calcium/calmodulin complex activates the enzyme called myosin light chain kinase, or MLK, by exposing its active site. MLCK phosphorylates the regulatory light chain on the myosin molecule. This phosphorylation increases the binding affinity of myosin to actin. This phosphorylation step requires a molecule of ATP distinct from the ATP required for the myosin head's cocking. When we talk about cross-bridge cycling within smooth muscle, with increased binding affinity, the myosin heads can bind to actin and engage in cross-bridge cycling powered by ATP hydrolysis, similar to skeletal muscle. This leads to the sliding filaments and muscle shortening. Smooth muscle can exhibit rhythmic or phasic or sustained/tonic contractions. The duration of the intracellular calcium increase influences whether the contraction is phasic or tonic. Tonic contractions can involve a latch state. In the latch state, the phosphorylated myosin head remains attached to the actin for prolonged periods, allowing sustained tension with reduced ATP consumption. Relaxation occurs when the myosin light chain is dephosphorylated by myosin light chain phosphate, which MLCP. The activity of MLCP is regulated by Rho kinase, which inactivates the MLCP when activated. This cell signals that inhibit Rho kinase allow MLCP to be active, leading to relaxation. So now we're going to compare and contrast skeletal muscle versus smooth muscle. Starting with the structure of both, with skeletal muscle, it's striated due to the organization of the sarcomeres. It has long cylindrical fibers or cells. It's multinucleated connective tissue sheaths. It's well-developed T-tubules and sarcoplasmic reticulum, sarcomeres that have distinct Z lines, A bands, I bands, H zones, and M lines. For smooth muscle structure, it's non-striated due to less organized myofilaments. It's spindle-shaped cells. It has a single nucleus. It's found lining hollow organs, not organized into large muscles with skeletal sheaths. It lacks T-tubules, less extensive sarcoplasmic reticulum, and myofilaments in sarcomeric arrangements, but less uniformly aligned, so it has less visible striations. When we talk about function of skeletal muscle, it's voluntary control, faster contraction speed. It shortens by about 30% of its own length. It can fatigue. It uses a sliding filament model. It exhibits isotonic and isometric contractions. Force generation influenced by motor unit recruitment, frequency, and length. When we talk about function of smooth muscle, it's involuntary. It has slower contraction speeds. It shortens up to 80% of its own length. It's very fatigue resistant, so it can maintain contraction for long periods. It also uses sliding filament mechanism. It exhibits phasic and tonic contractions. Force generation influenced by calcium levels, MLCK, MLCP activity, and latch state. When we talk about regulation of skeletal muscle, it requires stimulation by somatic alpha neurons. Its neurotransmitter is acetylcholine. Contraction is initiated by calcium binding to troponin. Primary calcium source is intracellular. Relaxation is via sarcopumps, and it lacks significant local regulation noted. For regulation in smooth muscle, it may be stimulated by your autonomic nervous system, neurons, hormones, intrinsic activity. Multi-unit relies solely on extrinsic. It uses various neurotransmitters, acetylcholine, norepinephrine, epinephrine, and hormones, either adrenergic or muscarinic receptors. Contraction is initiated by calcium binding to calmodulin, activating MLCK. Calcium source is both intracellular and extracellular for smooth muscle. Relaxation is via your MLCP, dephosphorylating myosin light chain. Calcium is pumped back into the SR with Circa pumps and exchanged out through a sodium calcium exchanger. It exhibits a myogeneric response, contraction to stretch, and pancrean regulation. Those are the main differences that we're going to talk about between smooth muscle and skeletal muscle. The last thing we're going to talk about in this week's video is muscle sensory receptors. The main two types are muscle spindles and Golgi tendon organs. So somatic reflexes, which control skeletal muscle, involve specialized sensory receptors called proprioceptors. Proprioceptors monitor the position of limbs, body movement, and strain on the musculoskeletal system. Two important types are muscle spindles and Golgi tendon organs. Muscle spindles, these are specialized proprioceptors that monitor muscle length. They consist of bundles of modified skeletal muscle fibers called intrafusal fibers, which run parallel to the regular contractile muscle fibers. Muscle spindles have extensive sensory and motor innervation. There are two types of intrafusal fibers, nuclear bag fibers and nuclear chain fibers. They do sensory innervation and motor innervation. Starting with sensory innervation, muscle spindles are innervated by large, fast-conducting myelinated group 1A fibers and smaller myelinated group 2 fibers. Stretching a muscle spindle activates these sensory fibers, leading to stimulation of an alpha motor neuron in the spinal cord. Motor innervation. Muscle spindles are also innervated by smaller gamma motor neurons, which activate the contractile ends of the spindles. This gamma stimulation helps maintain the sensitivity of the muscle spindle across a wide range of muscle lengths. This is achieved through alpha-gamma coactivation, where gamma motor neurons activate the spindle at the same time alpha motor neurons activate the extrafusal fibers, preventing the spindle from becoming slack when the muscle shortens and maintaining tension in the spindle's central region. This constant low-level activation contributes to muscle tone. The function: muscle spindles detect changes in muscle length and at the rate that it is changing. When we talk about Golgi tendon organs, these are also identified as proprioceptors involved in somatic reflexes. There's not a ton we need to know about them, but we just need to know that they are a thing. So with those included, we're going to talk about a couple of reflexes here. Our first one is a stretch reflex. This reflex is initiated when a muscle spindle detects a stretch. The sensory neuron from that stretched muscle spindle conducts an action potential into the spinal cord. Within the spinal cord, the sensory neuron directly stimulates an alpha motor neuron that innervates the same muscle that was stretched, causing it to contract. The sensory neuron also synapses with an interneuron that inhibits the alpha motor neuron supplying the antagonist muscle, causing it to relax. This is known as the reciprocal inhibition. The stretch reflex helps to maintain muscle length and tone. The other reflex that we have is withdrawal reflex. This is when you touch something hot, step on something sharp, and your body automatically compensates to overcome that.